We can just keep an eye on the time for the next few ones for the presentation. Hopefully that'll give us a little more leeway when it comes to the Q&A time. So the next team up is Team IllumiRNA, coming from the Anderson, Langer, and Sharp labs.
So it's a pleasure to introduce this interesting and exciting proposal. It is truly convergent. Dan Anderson, Bob Langer, and my lab. This is the second show. It addresses a very novel area of science, and that's microRNA and its role in cancer. And it addresses a technology to do something that is, as far as I know, never been done, and that is to sequence microRNAs from single cells. And it's a project that came out of discussions between the two labs, and it's going to be presented today by Salil Gar, and who's a resident pathologist and fellow here at the Koch in my lab, and Suman Bose, who's an engineer in Dan Anderson's lab. So Salil.
Thank you very much, Phil. And I couldn't agree more. I think this is the most exciting technology out of the lab since the split gene. So I'm here to tell you about our idea for early detection of leukemia through single-cell microRNA profiling. And to do so, we're going to start with the story of Jane, a hypothetical patient who comes into the clinic, and unfortunately, Jane is sick. She has flu-like symptoms, abnormal bleeding and bruising, and she gets a workup. And as part of that workup, she has a blood count test, which unfortunately for Jane reveals that 75% of her blood cells are abnormal. After we go through some of our workup in pathology, we realize Jane is diagnosed with leukemia. Now, Jane, unfortunately, has a very difficult prognosis ahead. She's looking at difficult combination chemotherapy with all the associated morbidities. She only has about a 60% chance of 5-year survival and a much lower chance of survival after that. But Jane came in 2015 for her routine physical, and as part of that, she also had a complete blood count test. And what we didn't know then, but she had a few pre-leukemic cells in her blood, but the complete blood count test can't find these because they're rare. They don't throw off the overall count. Wouldn't it have been great if we had a way to know back in 2015 that Jane was on her way to developing leukemia? We'd have totally different treatment options, and she may have a totally different prognosis. But there's a problem. There are no early detection tests for rare single leukemic cells. So our team has set out to solve this problem, and we're going to tell you about our vision, IllumiRNA, for single-cell microRNA sequencing. Now, I should say others have addressed this problem, including a sequencing company in the Bay Area that clearly stole our name. But they're going after genomic DNA in populations, and unfortunately, that's not the best analyte, because normal patients who will never go on to develop leukemia also carry mutations in genomic DNA. That's been shown now from a number of labs, including Ben Ebert's. So then we started to say, well, what biomolecule can we detect to identify these single rare leukemic cells? And we quickly hit upon microRNAs, the perfect biomarker and the emperor of all bioanalytes. And this is really because small changes in microRNA levels can change the function of thousands of genes, because microRNAs bind thousands of genes. That makes microRNAs and the microRNA profile of the cell a microcosm of what the cell is actually going to do. And in fact, totally different microRNAs are expressed in leukemic blood as compared to normal patients. So this is data compiled by our team from the publicly available Cancer Genome Atlas. And we can see for the top 30 microRNAs expressed in normal patients, they're expressed at very different levels with a very different profile in patients with acute myeloid leukemia. In fact, if we take just the top expressed microRNAs in populations of cells from normal people versus patients with leukemia, there's very little overlap. So this is a distinct profile at the population level. But we want to find that rare cell that was present in Jane back in 2015. So what are the technical challenges to do this? Well, if you think about it, we have to separate that population into single individual cells. Next, we need to isolate microRNAs from the other biomolecules that we're not interested in. And third, we have to have some way to tag onto those microRNAs a marker that we can then go back and say, these microRNAs came from this specific cell. So to tell you about how our technology platform does that, I'm going to turn things over to Suman. Suman?
All right, thanks, Alil. So as he pointed out, these are the really three challenges we have to address when we want to solve the problem. So we set out, or we designed our platform that does all of these three goals very efficiently, very fast, and in a very short time. So let me walk you through how does the device really work. It's a microfluidic device. And so in principle, what it does is it takes single cells and it encapsulates it within a droplet and then processes all of that cell within the same droplet. There is no mixing within the droplet, so essentially you are doing everything in a single cell fashion. So here you can see the animation, the device working. So what else goes within the droplets? We put capture beads. These beads are immunoaffinity beads that binds to microRNAs and will help us purify the microRNAs. And we put barcoding beads. This carries the little pieces of DNA that has the barcode that when I put on the microRNA will tell me which cell it came from. Right. So on the right, you can see this device in operation. It processes these cells at an enormously fast rate. This is slowed down 100 times. So we are encapsulating cells almost 50 to 100 cells a second, means you can process a milliliter of blood within minutes. So once the droplets form, the whole processing starts immediately. So the cells get lysed because of a lysis agent, and all of the microRNA and the bunch of other mRNA get released in the solution. And because there's a lot of mixing that goes on within these beads, the capture beads can really move around and capture all of the microRNAs in the solution. So here's a video at the bottom of an actual droplet showing you the mixing process going on within droplets. So within minutes, you have actually almost captured 80 to 90% of your microRNA within those droplets. Now, we still have one problem, right? We have a bunch of the other RNA we do not need and will interfere with my reaction. So how do we remove it? So there, the droplets arrive at the third station in our device, the T-junction here, where we have a magnet sitting. It's a programmable magnet that turns on whenever a droplet arrives. It pulls out this magnetic captured microRNAs along with the barcoding bead into a tiny little droplet and leaves everything behind. So that's really the key piece of technology allowing us to purify it. You could see this part working in here. A droplet arrives at the station. This is the magnet. It'll turn on. Boom, you have captured all of your beads, and then the droplet just goes away, leaving behind a concentrated sample. So you could do it very fast. Everything is in line. You don't have to interfere with the process. And this allows us at the end to get droplets with individual microRNAs from a single cell along with the barcoding bead. And the rest of the process is simple. You heat up the drops, the microRNA gets released, and then a ligase is used to tag these microRNAs with the DNA barcodes. Now, once I've completed this, the job is essentially done. I don't need the droplets. We can break the droplets. We can collect everything together, and then we do the sequencing. So let me tell you, how does it change how we analyze the data? So normally, say you have a bunch of normal cells, and you have a tiny one leukemia cell hiding in that bunch. You mash up the cells, and you isolate all the microRNAs, and you sequence it. You will probably not be able to identify the signatures of the leukemia because there's too much of biological noise. But now, because we have processed it in droplets, it allowed us to add these codes, the DNA barcodes, which tells me which cell each of the microRNA come from. And now I'm not going to look at the data in an aggregated fashion. I'm going to look at group as how are the cells processing this microRNA as individual cells, and then the leukemia signature will become very, very prominent. And that's really the key piece that will help us identify these leukemia cells within a bunch of normal cells. So let's go back to our resident pathologist to tell us how the story of Jane would change if we had this test.
Thank you, Suman. So now in 2015, things are very different for Jane. She gets the IllumiRNA, trademark pending, test, and we identify these rare cells because they profile completely different. We can get her into treatment, and she likely has a much better prognosis. Of course, we could use this for monitoring patients during treatment. And in fact, patients who have had leukemia and been treated, we could monitor them for remission. And that would actually be a great population to validate our test on. Right now, those patients are really just monitored for symptoms and by complete blood count, which, by our estimates, is about 1,000-fold less sensitive than our techniques will end up being. Now, we want to highlight one other aspect of this, which is, as Suman mentioned, these are immunoaffinity capture beads that circulate within the droplets. So you can actually use this platform, then, to do any technique at the single cell level that depends on immunoaffinity capture. Anything you do with an antibody. So you can do single-cell ChIP-seq, RIP-seq, proteomics, transcriptomics. This really could open up a whole new frontier, both in the clinic and for biology, really knocking down the Area 51 door of cancer and leaving precious few questions for Agents Scully and Mulder to answer. So our goal for the first year funding period is to construct the device, validate it on pedigree cell lines, and just see what's the variation even in cell lines among single-cell microRNA profiles. I think that will give us preliminary data to submit for other potential funding to carry the work forward beyond a year. So we have to acknowledge the team members. This has been a fantastic team just to be a part of, even in this brief time, with expertise across bioengineering, mechanical engineering, microRNAs in cancer, data analysis and deconvolution, and of course, the senior-most wizards who, without whose guidance, none of this would be possible. And oftentimes, our senior wizards have other places to be, so we work very hard to get their attention. So with that, I'd like to invite the other team members onto the stage, and we'd love to take any questions that the judges might have.
Okay, so thank you for a great presentation. That was very exciting. I guess as we think about this, my mind went to translatability. How do we translate this into the clinic? Are you creating a device that can be, sent out and used in labs around the country? And are you thinking about it? I wondered how we'd screen everybody. How expensive is this? And I guess the third question, well, I'll ask that later. Let's start with those two.
So first, in terms of the cost of the device is actually is very inexpensive. It's about $2 to produce. The sequencing is where most of the cost will come from. And currently, we only need to sequence about 10 million reads, so we estimate the sequencing cost itself will be about $40 with current sequencing technologies. So if we use the HiSeq, for example. And then in terms of actual translatability, we think our initial beachhead market, we're going to target people that, so for AML patients, for example, 10% of people that have had AML are likely to relapse, so we can initially start with them, and then we can move on towards other potential indications as well. So.
Yeah, and just to follow up, so we'll start with a diagnostic pool for AML, but eventually we could envision this as part of the hematology analyzer. And so for samples that are flagged as coming from physical exams, a portion diverts into our microfluidic device, which captures Jane cells back in 2015 and pulls out patients.
Did you want to make a comment? No. The next question I had on my mind is, can we get to solid tumors in any way with circulating tumor cells or anything like that?
Right. So it's been well established that microRNA profiles for solid tumors are also very distinct from, for example, the normal blood cells microRNA profiles. So you could use this as a platform for detecting circulating tumor cells from the blood using exactly the same approach that we described for blood cancers. You would simply then look for those individual cells with solid tumor microRNA signatures instead.
Colleagues, any?
Yeah, I had a question. It seems like a platform in a good way. And clearly leukemia is an important disease in treating that. But had you thought about the other applications beyond leukemia and a little bit with the solid tumor? How do you affect those other diseases? Because this could be quite broad, right?
Yes, absolutely. So this very much is a platform, and to us, that's absolutely the exciting part. This is obviously a cancer institute, and we're focused on cancer, and we think leukemia is a good place to put our foot in the door. But you can imagine any population of single cells now can be profiled for microRNAs. So we can monitor red blood cell diseases. Perhaps microRNA profiles are a better way to predict when a patient sickles. I think that's completely unknown, and so we'll open up new frontiers for diagnosis for other diseases and even just understanding their basic pathophysiology.
Nubar?
Yeah, I don't have even a CPA, but I'm going to ask a simple technical question. I mean, your example made it look like there's 40 cells and one of them was leukemic, but obviously there's millions of cells. So is the need for single cell just a way to get high sensitivity? And if that's all, versus really single cell, and if it's about high sensitivity, how else could you capture a small amount of cells and actually get to the microRNA? Because I can understand that single cell could give you information about the variability within the cell population, but you can't really act, I assume, therapeutically on that variability. And so relatedly, would you do anything different if you found one cell versus 100? Wouldn't you just treat it anyway? That's, I guess, the same question, but just what am I missing in terms of the single-celledness of it?
Yeah, so just starting to think about the current test. So if you had a CBC and you had maybe 1% blast cells or otherwise 10% abnormal cells in that complete blood count, you would suspect leukemia. So thinking about our test, based on our data for microRNA profiles for leukemic cells versus normal cells, we think we would only need to make about 100 reads, 100 microRNA reads in order to detect something. We could read about 100,000 cells then through our device and get 10 million sequencing reads total, as Tony's described. So if we could say, one of those 100,000 cells was leukemic or likely to be leukemic, and we could act on that, that would mean we're already at least 1,000-fold better than the current test. But let's say there is a lot of variability in microRNA, and that's something we need to find out since there's never been a single-cell microRNA sequencing technology available. If there is a lot of variability, within the leukemic population or within the normal cell population, let's say we could only really classify someone as possibly leukemic with 100 cells out of 100,000 or even 1,000 cells. We're still doing better than the existing test as far as sensitivity by 100-fold or so.
And if I could just add one point, I think you're absolutely right. Single cell is actually a great way to improve the sensitivity of your test. So for certain cancers like chronic myelogenous leukemia, where there's a stereotyped translocation, you could absolutely do this by RT-PCR and other techniques. But in diseases like acute myeloid leukemia and solid tumors, it's really a different derangement every time. So you really have to get down to the single cell level to see what the abnormalities are. I think ultimately cancer really is a disease of single cells going awry. And so we have to meet it at the single cell level.
Thank you guys so much. All right, we have people taller than me walking in front of me.